From Imperial College London, welcome to this special lecture podcast.
What I will try and do is to talk as much as I can about the relationship between medical imaging and mathematical modelling, but particularly in the context of the IUPS Physiome project. IUPS is the International Union of Physiological Sciences. And the work I'm going to talk about is very much the work from the Auckland Bioengineering Institute, with a lot of collaboration with other people around the world, but especially with Oxford University. The Maurice Wilkins Centre is a centre where we collaborate with people at the proteomics, genomics end of the spectrum. So what I'll do is first of all talk about the challenges of multi-scale modelling, and then go through the organ system that I've done the most work on and is perhaps the best demonstration at the moment of trying to bridge spatial scales from proteins up to whole heart function, whole organ function. But I'll talk a little bit about some other projects in Auckland, at the Institute in Auckland, in particular the lungs and the musculoskeletal system. And I'll also talk a little bit about the framework that we're trying to establish to handle the complexity at these different scales and dealing with the multiple physics that go into understanding function and structure across these scales. So first of all, the challenge of multi-scale modelling, and I think this is where I'd really like to emphasise the fact that just as we've had a revolution in genomics, our ability to now know the sequence of the human genome and rapidly acquiring a lot of information about transcription and the proteome, I think equally there's been just as much of a revolution at the medical physiology end in terms of imaging, which is allowing us to look in on the structure and function of the body. CT, obviously the fantastic advances starting with Sir Godfrey Hounsfield in terms of collimated X-ray imaging, but also MR, etc., that allows us to look at in PET and ultrasound and electrical imaging, all sorts of ways of looking at the function of the body in an individual patient context. And the challenge is, how do we link what we can measure at the genomic end of the scale with what we can measure for an individual at the whole body end in a clinical setting? And how do we develop a framework to handle the complexity that underlies going from 25,000 genes up to a single body? And with only 200 cell types, by the way, only approximately 200 cell types in the body. So here are the challenges. We've got spatial scales that go from nanometers to meters, a range of 10 to the 9. We've got temporal scales, even greater, from microseconds or less, up to human lifetime turnover of protein, 10 to the 15. And we've got a diversity of animal models that we use for understanding structure and function, from bacterial models for structural biology through mice and rats for functional genomics, through pigs, dogs, etc., large animals for a lot of physiological measurements, and of course applying to human with the clinical imaging technologies. And that's never going to be an attempt to devise a mathematical framework for understanding across these spatial scales. Is never going to be one model. It's a hierarchy of models where physics and engineering come in terms of partial differential equations at this end, but have to be related to the function at the protein level with systems of ordinary differential equations, often involving stochastic models, and as much as possible trying to link down to models of molecular dynamics and coarse-grained models of protein function to understand the measurements that we make at the single channel level with patch clamp, for example, techniques. And then pathway models for gene regulation and signal transduction, metabolic models. So there's a whole hierarchy of models where the most difficult challenge is how you bridge across these spatial scales. How do you interpret the parameters of a continuum model based on partial differential equations in terms of a more detailed knowledge of structure at a level below that. And I'll try and give some examples of where of strategies for bridging these spatial scales. So one of the things we're trying to do is to create a hierarchy of databases to deal with structure and function at these different scales, in the same way that the bioinformatics community has made web-accessible databases for the genome and proteome, etc., available. We need to do the same for physiology, and to base the encapsulation of models at these different levels in terms of XML markup languages, so that we can have a robust common framework for being able to deal with complexity by combining models that have been developed by different groups. So I'll talk later about this in particular, CellML, but a lot of the work in the Physiome project is developing this framework for handling a robust way of encapsulating mathematical models so that they can be used in a multi-scale sense. So here are some of the, at the organ level, some of the models that are being developed of the heart, the lungs, musculoskeletal system, and there's 12 organ systems in the body. These top 4 are the currently most developed, but rapidly many groups around the world starting to build Physiome-style models for the other organ systems. And all of these models use a common framework of, signal transduction systems turn up again and again in different cell types. A lot of the mathematical techniques developed for one organ system are equally applicable to another. And so there's a great deal of commonality in how we can address structure-function relations across all these organ systems. But I just wanted to also add in a reminder that we're only just beginning on this process. Although we've made quite good progress on encapsulating structure and function at the organ level for a number of organ systems, the reality is that we're only just beginning in terms of being able to handle the complexity of even the geometry of tissues and cells. And this is just a reminder that there are 200 cell types. They've all got their own organelle structure. You can't regard them as a homogeneous bag of chemicals. The spatial aspects of signaling within cells is certainly extremely important. And I think a rather nice point is that Sidney Brenner, who's one of the fathers of molecular biology, has always advocated that the cell is the key organisational unit in physiology or in biology. And we need to really address the complexity that exists geometrically at the cell level just as much as we need to deal with it at the whole organ and tissue level. And we've only just begun on this. We're many, many years away from being able to handle this level of complexity. But I'll show you a little bit in terms of myocytes. So here's the Heart Physiome project, and the idea is to be able to devise mathematical representations of structure and function at these different spatial scales. I'd say most of the effort from the Auckland group in particular has been at the level of organ and tissue, making use of lots of models that are developed by many groups around the world in terms of cell function, and linking in some cases down to protein function, but still a long way away from being able to handle the complexity of gene regulation that's vital in many diseases, understanding the changes of gene regulation that occur in many diseases. But nevertheless, beginning to set up a framework that allows us to bridge these spatial scales and figure out how to develop models where you can link across the scales, which I'll try and show you. So here's an illustration of what we're trying to do. Here's a model of the beating heart where the mechanics is modelled, and also the normal ECG for an electrical propagation. And then this is what can happen, can go wrong when, in this case, there's a particular mutation of a sodium channel that leads to a reentrant arrhythmia. This is a model that's been validated at the individual protein level and cell level, but not yet at the whole organ level. So as much effort has to go into validation as goes into developing the models, and also developing the instrumentation to make the measurements to do the validation. So I just want to point out that this particular mutation has not been validated yet at the whole heart level. But it illustrates what we're trying to do, to be able to link what happens at a protein level following through in its integrated cell behaviour, tissue behaviour, and then whole organ behaviour, and then out to the body surface where you can make clinical measurements. So the work I'm going to talk about is a large group of people. These are the people in Auckland. the work I've done over many years has been in collaboration with Bruce Smale, who actually did his PhD at Imperial College. And Bruce and I have worked together now for 25 years on aspects of the heart and other organs. And a number of our students have now become, many of them, lecturers. Nick Smith is now a lecturer at Oxford, are also very much involved in this project, and their students also. So there's a long history of people in the work that I'm going to show you. And I'm going to show you about first of all, the geometry and fibrous sheet structure of the heart, then how that's used for understanding mechanics, myocardial activation, the coupling of those processes, and then a little bit about the complexity at the cell level that needs to be dealt with in order to understand function at the larger scale. So first of all, geometry and structure at the organ level. This is just really a reminder of the extraordinary technologies, in this case an 11.7 Tesla MRI machine, that's available for revealing structure. This is an image obtained by Peter Cole and working with Nick Smith and others. But I think what hits me with these particular images is that our models are never going to be trying to capture a lot of the really detailed anatomical structure. There's an art to modeling. There's a process of making sure that you're not over-parameterizing or putting in too much detail, but that you can capture for the particular questions you're trying to answer, you can capture the essential detail that's part of the structure function for that particular question that's being asked. Because there's just no end to the detail that one could potentially include in these mathematical models. And the fact is that now we have access to extraordinary resolution images for this type of, at the organ level. And that data that I just showed, this is a student in Auckland and working between Auckland and Oxford, Jack Lee, who's been segmenting out the coronary vasculature from those images. So this is all done on an automated basis, so now it's possible to get very good reconstruction of coronary beds from, in this case, rabbit, from those MR images. So I want to talk a little bit about the mathematical framework behind a lot of what I'm going to talk about, and that is this idea of finite elements. And the idea, of course, many people here will be very familiar with it, but for those that aren't, the idea is that if you think of a region of tissue, very schematic here, but where you've got, say, a changing concentration of something that you're measuring, like the density, in this case, of GLUT3 in the lens, protein density distributions, and you want to be able to represent a spatial variation in that changing density of that protein. The way that you do it with finite elements is you divide the region up into small domains. You have parameters defined at the nodes of these elements that are giving you information about density that you then interpolate within a region to give you the mathematical definition of what's happening over this element. Similarly, you interpolate the 4 nodes here to give you the changing density pattern over this region, and so on. And then because the way that it's set up, these nodes that are shared along this common boundary mean that you get a continuous distribution occurring across the boundaries. And in fact, we often carry derivative information at these points so that you get not only continuity of the field, the protein density, but also the derivative of that field. So this idea of there being a continuum, a continuously varying spatial field that's used for defining both the structure of the underlying tissue and organ, but also the dependent variables that you're solving, which we'll come to in a minute. So here, for example, is the way that we set up the geometry of the heart. So we define the base, the 4 valves at the base of the heart, and these are using cubic Hermite elements, so there's derivative information carried. This is the endocardial surface of the right ventricle, and also including the muscle fibers. So these arrows here are the orientation of that, of those myocytes, and we measure that and capture that within the finite element framework as well, and both on the endocardial surface, the epicardial surface shown here, and then throughout the myocardium. So you end up with a mathematical parameterization of that variation in structure, fibrous structure of the heart, that you can then solve the physics on top of. And that detailed fibrous structure is critical both for the mechanics and electrical activity in the heart. So here's another view of that. These are the elements. And of course, when you're solving the physics on this, you may need to refine these elements to get a converged solution. But the initial mesh is set up in order to capture whatever degree of anatomical accuracy you need to capture. And this now has a translucent epicardial surface, just looking in on the inner endocardial surfaces of the right and left ventricles. So once you have that mathematical definition, we can now show the changing fibre orientations that have been measured and modelled with the finite element model, in this case showing the variation of fibre direction, which starts out as being about minus 60 degrees to the circumference on the outside, and as you go through the wall it becomes almost vertical on the inside. For those that were at the talk this afternoon, there were some beautiful images of fibrous structure in the heart. And one other aspect of the structure of the heart that is something we discovered a number of years ago, which is shown in the next slide, is the fact that those myocytes, the fibre orientation, is actually coupled together into sheets by the way the collagen is organised. So if you think of a small chunk of left ventricle taken out of the wall, this is the organisation of those myocyte, or the fibre direction changing across the wall. But those fibres are coupled together, what we discovered, and then discovered that other people had discovered it earlier than we had, particularly the German literature in the 40s. I don't think they realised the significance of it, but there's certainly reports of this organisation of the collagen from earlier work. So we rediscovered it and then realised the significance of it. And the significance is that you have to think within the heart both about an orientation of a dominant direction for the fibre along which the muscle contracts, but you also have to think about another vector which is at right angles to that, which defines the way those myocytes are tightly coupled together. And they're about, if you look at this figure here, those myocytes are coupled into sheets, 3 or 4 cells thick, and the sheets can relatively easily shear between, sort of slide, or not really slide, but shear in a continuum sense across these cleavage planes. So the myocardium is organised in an orthotropic fashion. It has a dominant direction of the fibre, another direction associated with the dominant binding of cells into these sheets, and that would be this direction here, and then a third orientation which is just at right angles to that in the reference state. And you see that both in gross microscopy, just very easy to see in ordinary microscopy, and in electron microscopy, and in confocal imaging. And we've done many, many studies now looking at the structure. So here are the individual cells coupled together into sheets. The sheets branch. This tissue has been opened up, a hypertonic solution to open it up to show them more. These are the capillaries, 1 capillary per myocyte. The capillaries align very closely with the orientation of the myocytes. So we now have a pretty good picture, I think, of the structure of tissue at this level of detail. Here's another view of the organisation of these sheets and the branching between the sheets that's occurring here, for example, and we call these cleavage planes, but the very distinct laminar organisation of this tissue. So we built a piece of equipment funded by the Wellcome Trust to try and automate the process of measuring the structure across spatial scales. Here's the machine, moves a block of tissue between a confocal microscope and a precision mill, and we can move across 4 orders of magnitude and build up a detailed picture of going from the whole heart cross-section down to individual myocytes, and look at the way the collagen is organised around those myocytes. So this equipment now is in constant use for looking at a whole range of soft tissue, but it was developed initially for cardiac tissue. So here, for example, is information pulled out of those tissue blocks where we're segmenting out the collagen structure from the reconstructed confocal 3D confocal images. So now we can see the organisation of the collagen, both its density and its orientation across the wall of the heart. Notice that there's much lower density in the central region of the heart, of the wall rather. You can see the organisation of the sheets and then the collagen relative to that sheet organisation. And then you could segment out the individual myocytes themselves. So this is now moving up through a block of tissue and having stain for collagen, which allows us to look at the way the paramesial collagen that binds the cells into the laminar sheets, how that moves across through the tissue. And then what we're doing is automatically segmenting these in order to build models that have this level of detail in them in order to then extract out an understanding of the mechanical properties for constitutive laws at the continuum level that you use with the whole heart. So let's look at mechanics then. We've now got a definition of the geometry of the heart, its organisation of its fibrous sheet structure, and some knowledge of the underlying tissue properties. So we want to use that for studying mechanics. So you take, mathematically, what we do is to have these finite elements which are defining the geometry. You can think of a mapping between real space here and a mathematical space here, where we're carrying this information, all sorts of information like the geometry, fibre sheet orientations, electrical potential, all the dependent variable fields for the particular equations we're solving at the nodes of these elements, and then it's tricubic Hermite interpolation, so you carry derivative information as well. So the idea is that once you've got this definition of the anatomy, then you can solve the physics for a whole variety of fields interpolated over these regions. So, for example, if you look at a small region of, or certain points within the myocardium, there's a dominant direction, which is the fibre direction, but then there's also that sheet organisation shown here by the subtle plane. So we need to define the mechanical and electrical properties of the tissue based on material axes which are aligned with these local fibre sheet orientations which vary everywhere around the heart. You can see the fanning across the heart wall of these myocytes and also the changing sheet orientations. So then we can set up equations that are based on looking at the undeformed geometry and the deformed geometry of the tissue, define base vectors tangent to the coordinates, define metric tensors that define the structure of the tissue in the undeformed and deformed case, and then set up strain tensors which we can use to define the deformation of the tissue, and then use those with information about the material stress-strain laws for the tissue, which I'll talk more about in a minute, and then we're just using the equations of motion coming from stress equilibrium, so conservation of momentum and conservation of mass. And so these are well-established techniques, Galerkin finite element methods. Perhaps the only unusual thing is this fairly complex definition of the material properties and the fact that we're using high-order finite elements to solve these equations. But otherwise, it's a pretty standard engineering approach to handling a large deformation continuum problem. So the key thing are the relationships between stress and strain and the definition of the strain energy functions which define the properties of the tissue. And we try and derive those to some extent from our knowledge of the tissue structure, but in the end we also have to make measurements directly on the tissue. And so what we do is we've built equipment to measure put a small piece of tissue into a rig where we can control the forces acting on the tissue, measure the deformation, and out of that extract the stress-strain behaviour of the tissue, which is highly nonlinear. So like all soft biological tissues, when you stretch it along one axis, so this is the strain axis, it very rapidly stiffens and you get a large increase in stress as you reach particularly the strain limit of the material. So we define a strain energy function that's orthotropic, different in three directions, and captures that nonlinear stress-strain property and is related to this organisation of the structure of the tissue at each point. So here's that bit of equipment in operation. You can see the pullers stretching the tissue. There's two axis force transducers on the end of each of these, and we image the deformation field with various techniques like speckle interferometry, and then extract the deformations and then back out the material properties using a model that takes account of the inhomogeneous structure of the tissue in order to extract stress-strain behaviour. And we also have equipment where we can do shear loading to measure shear properties of the tissue as well. Then you also have to have a knowledge of the active properties of the tissue. So we've built equipment to measure the behaviour of trabeculae and also particularly using the work of Hank de Keurs and many of his students, Hank de Keurs in Calgary, who's done many measurements on the active properties of cardiac tissue. So we set up models where we have the kinetics of calcium binding, tropomyosin kinetics, length dependence, and cross-bridge kinetics. I'll skip this, but just the fact that you end up with a system of integral differential equations defining the properties of that active material that can then go into the continuum models. So now we're in a position to look at predictive models of how the heart works as a pump. We know the geometry, we know the fibrous structure, we've measured material properties, and we can put all that together and predict based on the equations of motion what the stresses and strains are in the heart under certain boundary conditions, such as the ventricular pressure. I'll talk more later about pushing those boundary conditions further away by including fluid mechanics, but for these simulations we're using the pressure in the ventricles as our boundary conditions. And the fact that from a standard 1.5 Tesla MRI now it's possible to get really good data, and we use MRI for validation with controlled loading of animal hearts. So what we're in a position to do now is to predict how the heart works mechanically. And I think as well as the way to understand the deformations going on in the heart is, first of all, to observe that there's a great deal of shearing and twisting going on, which is partly a result of that changing fibre organisation across the wall of the heart. But a really important part of the mechanics of the heart is the fact that the sheet structure allows the sheets are organised in a radial fashion in the centre of the wall, and allow this large degree of base apex shortening that occurs because of the compliance in that third direction orthogonal to the sheets. But the sheets turn over to become around plus or minus 45 degrees in the subendocardium, and it's the shearing action that really allows the heart to pump effectively. So the very large wall thickening that you get right at systole, where you get that kick now that's a consequence of the material properties that allow the tissue to shear easily, and that's a consequence both of the fibre and the sheet organisation. Might be better shown in the next one. So here's another view of that, where now you can see depiction of myocytes and how they change their organisation across the wall. But if you look at the changing dimensions of the tissue, the wall thickening that's occurring during systole, now that huge kick is very much associated with the way that the tissue can shear. So it's a property of this laminar organisation of the tissue. And so we can look and see what happens in various diseases where you change the collagen density, such as hypertrophic cardiomyopathy, to see the effect of the material changes in material properties, how that affects its ability to shear and function as a pump. Here's another view of that, where a block of tissue now which starts off as a cube is now being elongated, stretched in various directions in diastole, and then if you watch that cross, that very large degree of shearing that's going on. When laptops were slower, it was easier to see this because it was slowed down a bit. But if you just watch particularly that kick at the end, these arrows are the principal strain vectors, ligand vectors. And this is just an end-on view into the myocardium, showing the shearing action going on in the other plane. So one application of these models is to clinical MR, and this is the work of Alistair Young, Brett Cowan in Auckland, where they use tagged MR to fit the models to patient MR data, and that is good for getting the kinematics of the heart. It doesn't tell you what the stresses are, but it means that you can, by fitting the models to the tagged MR data, you can look at the regional distribution of strain. But obviously you need pressure as well to get stress out. So all of that is switched on by a myocardial activation process, and thanks to the work of Dennis Noble in Oxford and others, but particularly led by Dennis from the early 60s, our understanding of cardiac electrophysiology is at a very high level in terms of understanding the individual ion channels. So we know that there's a fast sodium channel responsible for the upstroke of the action potential. There's a transient outward channel that gives this dip in the action potential. There's an L-type calcium channel that's responsible for triggering calcium release within the myocytes, which leads to contraction, and also because it ends up being a slightly inward current, supports the action potential. And then there's a bunch of, there's also a sodium-calcium exchange, also giving an inward current, raising the action potential, and there's a bunch of potassium channels that give the repolarization. So by understanding and measuring the proper time and voltage dependence of the gating processes for each of these channels, we can put all those together into composite models of cell function to predict action potential function and how that changes under different heart rates, etc. So the critical thing in this is how all of these channels end up affecting the QT interval, or the distance between the upstroke and some measure of the repolarization of these cells. And what we now can do is to include models that deal with all these ion channels within the tissue and whole organ context to look at the normal heartbeat, but also arrhythmias. And one of the complexities, one of the additional advances that's come in the last few years is the recognition that these models have to accommodate the stochastic behavior of many of these channels, because drug binding affects the probability of transition between these states. And now there are good models, for example, developed by the Ray Winslow group and others who are developing stochastic models that could also plug into this framework for looking at drug action. So here's an example of how we can use multi-scale approaches to deal with the problem of going from the tissue and cell level up to the whole organ level. So here's the block of tissue that I showed earlier from confocal imaging reconstruction of the organization of that tissue across the wall. And then we need to have that level of detail to understand how current flows around the cells. So now we can have, if we put that level of detail into a block of tissue and we use the ion channel models, we can look and see how if we stimulate at a particular point in the tissue here, we can watch the propagating action potential wavefronts, shown in red for the upstroke and then blue for the repolarization. And by including a lot of this detail at this level and solving reaction diffusion equations for the propagation of the currents through the tissue, we can then fit these models to much simpler models that just have an orthotropic conductivity tensor based on that fiber sheet organization that I showed before. And that means we're bridging scales because we don't need to handle this level of complexity for the whole heart. We can, from this, derive the simpler models that can then be used for understanding propagation in the whole heart. But the important point here is that we have to be able to define the conductivities associated with the different directions and associated with that fiber sheet structure. So then we can put that into models of the propagating action potential wavefronts around the whole heart, starting from the Purkinje fiber innovation. And then these models are now able to then start from the ion channels and go up to the whole propagation on the whole heart. One of the complexities I'll come to in a minute is the fact that the spatial organization, spatial density of proteins becomes quite critical for understanding reentrant arrhythmias. So what we want to do now is to couple those two things together. The activation process switches on the mechanics, releases calcium and turns on the whole contraction process. So we've got to deal with the numerical problems of coupling activation with mechanics. So as I've said, mechanics is solving large deformation elasticity equations to give from some initial configuration under boundary forces to give a prediction of deformed tissue, and then we can solve reaction diffusion equations for the high density of points needed to capture the reentrant currents that are going on in the myocardium. You need a much larger grid for capturing electrical activity than you do for mechanics. You need about a 0.1 millimeter spacing, so you end up with a very large number of points, but you can use techniques like multigrid to accelerate and only handle high density where you need it in the propagating action potential wavefronts. So the idea is you solve the mechanics, solve the reaction diffusion equation based on the deformed configuration where you've updated all the metric information about the conductivity tensors. solve reaction diffusion for voltage and producing calcium release and tension, feed that back into the mechanics, and then iterate around that loop until you get convergence. And what we now do is to code all the cell level equations in a language which I'll talk about in a minute called CellML, so that that gets automatically incorporated into the tissue organ level codes. And here's an illustration of the difference in numerical density required between mechanics and electrical activity. Here's the mesh for mechanics, for Solvit-Galerkin large deformation equations, and here's the mesh, and these are the Gauss points for quadrature, and these little green points are the points for solving the reaction diffusion problem. And as the heart deforms, those little green points get carried with it, but it is now a very large numerical problem to solve the reaction diffusion. So here's an illustration of that coupling between a propagating action potential wavefronts based on these ion channel models, leading to dimensional changes of the tissue as you get calcium release and mechanical activity. And that the red wave is the activation and the blue wave is the repolarization as the potential comes back down again. So we can look at potential, tension and length changes in a block of tissue. Or in this case, in a rather simplified heart, in this case, for the action potential wavefront leading to, you can see all the shearing action, which is rather exaggerated in this case because of the symmetry of the model. So one of the complexities that we have to deal with is the fact that, and we're only beginning to deal with this, is the fact that in particular the transient outward channel that I mentioned earlier is expressed at a much higher density on the epicardium than it is on the subendocardium. And so the epicardial action potential has got much more of a spike and dome shape to it than the endocardial waveform, which is more triangular. And that changing density of that particular protein and other proteins like IK1, for example, means that you get spatial variation in the repolarization that becomes critical for re-intraday arrhythmias. So we have to be able to understand the spatial variation of density of these channels in order to properly understand normal propagation and re-entrant arrhythmias. So that requires that we do mRNA analysis or ideally protein density analysis together with patch clamp techniques for phenotype to understand that spatial variation in the models, which is not only transmural but is also well known to be also occurring around the heart. I mean, there are many things like Brugada syndrome, for example, that are critically dependent on the changing spatial variation of some of these channels in the circumferential direction. Another aspect to the function of the heart is the coronaries, of course. This is the work of Nick Smith in Auckland, now in Oxford, in his PhD in Auckland. So modelling pulling out the segmented coronaries and solving Navier-Stokes equations down these vessels, at least in this case one-dimensional Navier-Stokes, where we assume a profile for the blood flow across and then couple in the deformation of the myocardium onto the pressure area relationship for these blood vessels, and then solve so that we can look at and see how during the deformation of the myocardium blood is being shunted around from subendocardium to epicardium. But this is a reasonably fast solution now because it's only one-dimensional Navier-Stokes, and it has the limitation that you're having to assume a particular velocity profile across the wall of across the radius of the blood vessels. And where the direction this work is going is to understand metabolic supply-demand relationships. So we want to know, for example, where the energy demands are highest associated with the large work loops, stress and strain behaviour at certain parts of the myocardium, how that impacts on the delivery of blood and metabolites to the oxygen metabolites to the tissue. But one of the factors which is not included adequately yet in these models is the control of smooth muscle control of the aperture of the vessel. So this is effectively an adenosine-treated heart where all the blood vessels are wide open. So there's another aspect to the control of those blood vessels which is not yet included in these models. And then another aspect is the fluid mechanics. We need to push the boundary conditions further out, ideally well away from both the entrance into the pulmonary veins or the exit well down the aorta. So we're dealing, as many groups of course here are also at Imperial, are solving the three-dimensional fluid mechanics using either finite volume techniques, AL techniques, or finite element techniques, or other techniques. But the challenge now is to link the 3D fluid mechanics into the wall, and I think we're going to learn a lot more about the mechanics of the heart once we have fully coupled fluid structure models, particularly where we're allowing the base of the heart to be free, and so we're pushing boundary conditions back around including the atria, which we're just starting to model now. But I think there's a lot to learn in terms of the interaction between the wall mechanics and the fluid that will, I'm sure, change our understanding of the whole nature of the mechanical function of the heart in the coming years. So I just want to push down now into the cell level processes more, because there's a great deal of complexity at the cell level that is not yet handled, that's quite important for understanding cardiac function. These are the areas that are critical. So we know a lot about electrophysiology. We know quite a lot, thanks to the work of Richard Vaughan-Jones at Oxford, about the proton bicarbonate transport. We know a lot about calcium transport in the heart, from many, many people working on calcium in the heart. And we know a lot about mechanics, myofilament mechanics. We're just beginning to get good models of metabolic pathways. We now have quite good models of beta-adrenergic pathways and CAM kinase pathways, and beginning on models of IP3, diacylglycerol, and some of the other pathways, but a long way to go really in terms of properly understanding some of the issues of signal transduction. And we're only just beginning in terms of gene regulation, which is going to be critical for a lot of disease processes. So here's some of the complexity that's currently in the models of cell function. The sodium-potassium channels, sodium-potassium pump exchanges and pumps, calcium, etc. Calcium transport processes, release of calcium from ranitidine receptors, uptake and circa pump, all that is quite well modelled and validated now. Buffering and contraction, and then the whole process of regulation of protons, pH control in the heart and bicarbonate control is coupled into the models. And we're beginning to couple in a lot of the metabolic pathways. So I'll talk shortly about the framework for handling that complexity, because you now have to combine the work of many groups around the world who are developing different aspects of the understanding of cell level function. But I just want to come back to this issue that it's really a three-dimensional cell. We're still treating it as a lumped bag of chemicals. It's not. It's got a highly organized structure. And this is just an illustration of doing two-dimensional reaction diffusion modeling at the Mark site using data from Mark Cannell and Christian Soller in Auckland, who used two-photon microscopy to give structure. And we're beginning to develop 3D models which can handle some of the issues of spatial variation within the cells, but we've got an awfully long way to go in terms of understanding the issues of spatial variation in cardiac function at the cell level. So I think I'm going to very briefly, and I'm going to run out of time, so it's going to be brief, talk about similar issues of combining imaging and modelling in terms of understanding lung and musculoskeletal function, and then very briefly this framework. So this work is led by Meryn Tarfai in Auckland, and she's modelling the blood vessels, the airways, and tissue mechanics, and coupling those together at the whole organ level, but also looking at gas exchange at the alveolar level, so building models of capillary networks around the alveoli, looking at gas exchange, and then linking that back up to the function at the whole lung level. So and she's working partly with a group at Iowa, Eric Hoffman at Iowa, using sheep lungs for the validation work. So building models where we can construct the airway trees, and then particularly look at, this is an illustration of one of the reasons why the interaction between modelling and imaging is important, that typically you image the lungs in prone or supine positions, but you want to look at function when it's gravity loaded in a vertical position, so you can fit the models to the imaging setup and then reconstruct the trees, but then you want to then turn it around into the vertical position, and then when we solve the airflow and blood flow and mechanics, you can then account for the fact that the fitted geometry in this position is now being properly accounted for gravity effects in the vertical position. That's just one illustration of why I think the use of modelling together with imaging is so critical. Another illustration, this is Kelly Burrows, who did her PhD on looking at blood flow in the pulmonary system, and just again illustrating this idea that you can use the functional imaging for model validation, but also help in understanding imaging measurements and for predicting function. But I don't think I've got time, and I'll very briefly talk about musculoskeletal. So here's models we've developed in Auckland for the musculoskeletal system, starting from the outside surface and going down to the bones. And this illustrates this process of fitting detailed data on the geometry of bone and also the structure in the same way that we were doing for the heart. And I'll show in a minute how we then adapt that for patient-specific geometry. So these models are now being used for looking at things like cerebral palsy, walking, abnormalities of walking, and a number of aspects of musculoskeletal function, where by modelling the anatomy and the structure of the tissue in a way that allows you to embed models down to the cell level within that environment. And this is just illustrating this process of how we go from, in this case, using the visible human data, where we can take high-resolution images to help create the generic models that we start with before applying them to patient data. So here's a process of segmenting out a particular muscle from a given imaging plane, and then doing that at a number of planes, constructing data that is then used to construct an initial linear finite element mesh for the outer surface of that muscle. And then by using these high-order cubic Hermite elements, fitting the data with these higher-order elements, we can get an efficient parametric description of that muscle shape and the structure of the tissue within that muscle. And then we can build up a database of these muscle geometries that we can then pull out of the database for looking at different particular applications. And then this is just showing that process of this generic database that has the reasonably accurate anatomical descriptions of all the bones and muscles in the body, then what we need to do is, once we've got these detailed models based on the visible human data set or other sources of information, we need to fit them to individual patients. And the way you do that is, if you have imaging data where you can start with the generic model that's been constructed, but now identify fiducial points in the patient image target and the corresponding points in the generic model, but embed that model inside a region of space that can then be with free-form deformation, or what we call host mesh fitting. We can then alter the geometry to fit the patient-specific information by effectively altering the space around that model is embedded in, to give us a customized model. Once you export that model, you've then got a finite element model that's tailored to that patient that you can then solve the mechanics on. So finally, just to talk a little bit about the framework for handling complexity at these different scales. And the strategy is to develop markup languages for encoding these models, develop libraries of open source tools, model repositories based on the markup languages, and then various environments for implementing collaborative projects. So the work here is the Auckland group, led by Paul Nielsen, and also working with people at Oxford and Sydney. And CellML is a markup, XML-based markup language description of cell function that encapsulates information about the model components, the mathematics that describe those processes, and then a lot of metadata described in RDF about the ontological information about the components of the models. And there's now about 350 models on the database, that's the site, and each of these models is encoded in a way that then you can automatically create the computational code to run the models. So here, for example, is doing a search on cell signaling, which pulls up about 40 models of cellular signaling, in this case a beta-adrenergic pathway model. And this is an interesting model because it's the first one that really couples signaling, in this case beta-adrenergic signaling, to phosphorylation of proteins that are involved in mechanics and electrical activity at the tissue and organ level. So we're starting to get a link between signal transduction and how that affects function at the organ level. So then the CellML framework allows you to see the mathematics, to render the mathematics automatically. We can also start to apply all sorts of tests where we want to make sure the models are validated in terms of matching the published paper, making sure that they have the added parameters to make sure they work if there's information missing from the paper, and then to apply other measures of how valid these models are in terms of the laws of physics. Do they obey thermodynamic constraints, etc. So I'll skip this. This is just the ontology for linking into biological databases. And then there's a bunch of tools that are open source tools being developed to allow these models to be run. And I will finish by summarising that what the Physiome project is trying to do is to link to clinical imaging, because that's the way of making sure we're developing models that are patient-specific, but use the power of continuum modelling using the constraints that are imposed by the laws of physics to interpret those, what we can obtain from clinical imaging in terms of function, and to base constitutive laws or description of material properties on detailed knowledge of tissue structure, and try and reach down as much as possible to protein level function, because that's where drugs operate and disease operates. And we're doing that within an open source software environment, developing standards for encoding models and establishing web accessible model databases for shared use of these models. And finally, the acknowledgements to funding sources, which are a number of sources within New Zealand, but also the Wellcome Trust, NIH grants, EPSRC, and one European grant. But also very much for the heart electrophysiology work with Dennis Noble, David Patterson, Peter Cole at Oxford, many other colleagues around the world, but most of all to our grads and postdocs who are behind the work that I've shown you. So thank you very much.
You've been listening to a special lecture podcast from Imperial College London.